Hi everyone, my name is Pratik Agarwal and today we are going to discuss about drug absorption in human body. So we are going to touch upon the topics like what are different types of drug absorption, what are the factors that affect it, and we'll touch bases upon bioavailability and bioequivalence topics. So first let's learn about what is drug absorption. The time when the drug is administered in our body to the time when it reaches our blood and gets distributed is known as the absorption. So when we swallow a drug or drink a drug in liquid form through oral route, it reaches our stomach and intestine. From that part onwards, the time it reaches to our blood is known as absorption, from where the blood distributes it to various parts of the body or also to the target organ. Now, what are the factors that are affecting this absorption of blood? The main factors usually include the of the environment, ionization constant of the drug, which we'll be discussing later. Now, there are different mechanisms through which drug is absorbed in our blood from our stomach or intestine. They are passive diffusion, facilitated diffusion, endocytosis, and active transportation of drug. Now, we'll learn about each of them in a bit of details in our future slides. So, let us learn about passive diffusion. The word it is self-explanatory that passive diffusion means that the drug is reaching or crossing the cell membrane and reaching the cells or without any efforts being taken. It is an automatic process, you can say. As you can see in the diagram, there is a region of higher concentration where the solute is more than the solvent, and there is a lower concentration where the solute is less as compared to the other compartment, and it is separated by a cell membrane which can be seen as a barrier between the two compartments. As we have learned earlier, in diffusion, passive diffusion, a concentration of higher gradient transfer the solids to a concentration of lower gradient separated by a barrier. So here the drug from outside the cell gets inside the cell by passing through these barriers known as cell membrane through the help of concentration gradient difference or passive diffusion. This continues until the concentration of the drug is equivalent or is in equilibrium on both sides of the cell membrane. So this is an automatic process. Now, in this process of passive diffusion, if the drug is water soluble, then it has to transfer through water channels or pores that are constructed within the cell membrane. But as the nature of the cell membrane is lipid based, we also call cell membrane as lipid bilayer, made up of phosphate as well. So lipid soluble drugs can easily transfer through such cell membrane barriers. So that's a bit of difference, but otherwise this is how passive diffusion occurs. Now let us learn about facilitated diffusion. Again, the word itself is self-explanatory. What does facilitated means? It means that it is being assisted by something. The transfer of drug from an extracellular environment to inside the cell in the cytoplasm is being facilitated by something, is being assisted by something, and it is being assisted by these large protein molecules, also known as carrier proteins. Now, these carrier proteins attach the drug to itself and forms a passage that helps in transfer inside the cytoplasm. Sometimes we refer to as transport RNA. Transport RNA transports the drug sometimes inside the cells. We can take that as an example to understand that how facilitated diffusion happens, how these pores or assistance takes place to help drugs from move from an extracellular environment to an intracellular environment. Now comes up the active transport. Now this active transport, cannot be understood by itself through the word that it says active transport, but let us assume that, as we said, there was a two different concentration gradient separated by a barrier known as cell membrane. In passive diffusion, the high solutes from higher concentration gradient move to the lower. But in active transportation, we transfer the solutes from lower concentration gradient compartment to compartment with higher concentration gradient. So, for example, separated by a cell membrane, a barrier, there are two compartments outside the cell, inside the cells. Now, inside the cell, the concentration of the drug molecule is already high as compared to the outside of the cell. But still, we need the drug to be transferred inside the cell through the cell membrane. Then, active transportation takes place where extra energy is deployed to transfer the drug from lower concentration to higher concentration with the help of carrier proteins. Now, this happens because there is a molecule in our body which provides us energy, also known as adenosine triphosphate. When this molecule is hydrolyzed, it transfers a phosphate group in terms of energy and carries out all the processes in our body, one of which is active transportation. This molecule ATP is then converted to ADP, adenosine diphosphate. And the last process is known as endocytosis. What does endocytosis do is it opens, like for example, the cell membrane opens, forms a vesicle and engulfs the drug from the extracellular environment. Then it closes this vesicle from the outer region and pinches off the drug inside the cytoplasm by opening the other part of the vesicle form. This can happen vice versa, like the drug might be transferred from inside the cell to outside the cell, and this process is then known as exocytosis. So I hope you were able to understand that what are the four different forms to which drug is transferred or absorbed from the outer region to inside the cells. Now, let us understand what are the factors that influence this absorption. As mentioned, pH and ionization of drug is one of the important factors. Now, for example, there is a acidic and a basic drug. Acidic weak acids are already present as learned in an charged form, but in an uncharged form. But basic drugs or weak basics are present in a charged form. When ionization occurs, the acid donates a proton, an electron, and it becomes charged. But on the other hand, when ionization occurs of a weak acid, it donates its proton which carries a charge and the drug becomes uncharged. Whenever absorption happens, whenever a molecule needs to go through a cell membrane, it can easily go through what you say uncharged form. So if weak acid isn't ionized, then it can pass through the cell membranes easily, whereas weak basic cannot. But if ionized, weak acids cannot, but weak bases can because it has donated the charged proton already. You got my point here, I hope so. Now, if it is charged, if it is ionized, for example, we can say, how would they pass? They can go through active transportation. It is then when active transportation comes into place when we have to forcefully transfer drug. It's not always used when we have to go against the concentration gradient. It can use in such instances also. I hope you understand. Now, blood flow. We obviously know that in order to get the drug to our target organ, we need to distribute it from our stomach to the target organ. Now, with the help of absorption, we were able to transfer the drug into the blood, and then the blood needs to distribute to every part of the body because drug needs to get absorbed in the cells, and then cells throws them out and get them absorbed in the right tissue. Okay, so blood flow is more at intestine as compared to stomach, and that is why most of the drugs get absorbed in a small intestine. So if we are trying to get our drug absorbed from a region of low blood flow, the absorption rate would be less. But if we get trying to get a drug absorbed from a region of high blood flow at intestine, then the absorption rate would be high. Similarly, surface area. The drug needs a lot of surface area to get absorbed. In our body, small intestine has the most amount of surface area. Why? Because of the villi. Our small intestine is like folds. So it's got thousands of folds as we have learned earlier in school or colleges. And these contains villi, which transports the drugs as well forward. So as this has a lot of surface area, this adds another point that why most of the drugs gets absorbed from the small intestine. So if we try to get a drug absorbed from an area of small surface in a region of small surface area, then the absorption rate would be low, less chances. But on the other hand, if we try to get them absorbed from a region of high surface area, then there would be more chances of a drug getting absorbed. Contact time. Contact time is a very tricky thing, a very tricky factor that influences our drug absorption. If there is not a right amount of contact time, then the drug absorption would not be adhered. It would be effective. Let us say that we got a problem. Let's say I've got vomiting or diarrhea. Now, if I take a tablet of any medication and I get diarrhea, then that drug from my stomach would pass and excrete without being absorbed, without being transported into the target organ, and it is not going to show its effect. So it would be ineffective. No absorption is going to take place. Similarly, if I vomit it out, then all the contents of my stomach are out of my body, including the drug I ingulged. So again, it would take time, or it wouldn't like get absorbed in a body and wouldn't show its effect. So the contact time should be more in this case. Let's say in the case of diarrhea, we're getting less contact time. But for example, I take a drug and I eat it with food. Now, in my stomach, I got food and drug, both of them. Now, the food will take time to pass through the passage and go into the intestine and get absorbed in my blood as a nutrients and transport. And because it's going to take time, it's going to be attacked by a lot of acids that destroy the food to extract nutrients from it. With that food is my drug, which might be acted upon acid. That's a different thing. But because there's a lot of food in my stomach, my drug is taking time to get past to the stomach area and go into the intestine where blood flow is more, surface area is more, to get absorbed in my blood. So as it is taking time, it is getting attacked by the acids that are produced in our stomach, like Pepsi. As it is getting attacked, it is getting destroyed. The active ingredient is getting destroyed. More time is taking place. Till the time it reaches intestine after the food or with the food, most of the active ingredient has been destroyed. And there is very less, or we can say not enough amount of drug left to be absorbed from intestine. Hence, we do not get good results from the drug that have been taken. So that is also one of the reasons. One, the last reason that I want to talk about in depth is P glycoproteins. These are also a membrane protein. How does it help? It is responsible to pump drug out of the cells. So what it does, it is present on various tissues. It's not present all over body, but it is present in various tissues. And what it does, it takes the drug, throws it out in the what you say blood cells. We don't know how, we don't know why, we don't have that scope, but we know that this is the reason why there is multi-drug resistance. That is not a major reason, but it is involved in the multi-drug resistance. Other factors that can affect the absorption of molecules, drug molecules, are disintegration. Our drugs needs to be simplified, like it needs to be disintegrated or soluble in the environment that it is getting absorbed from for an easy absorption. You eat a tablet, the tablet itself cannot get absorbed from the intestine, so it needs to break down. So the disintegration factor or the dissolution factor of the drug also plays an important part in getting absorbed. So these are some of the factors that affects the absorption of drug. Now, as we have learned about different types of mechanism through which drugs are involved and the factors affecting it, we'll touch bases upon the last topic of our lecture, which is bioavailability. We all know that bioavailability is the extent to which our drug is absorbed or the amount of drug that is received to the target organ in comparison to systemic circulation. Because if I take parenteral routes of drug through systemic circulation, 100% drug is reaching because it's not getting attacked by any acids, it's not getting metabolized. But if I take the drug through oral route, then it is going to get in my stomach, get attacked by acids, going to absorb. Not all drug is going to get absorbed due to factors mentioned and then get distributed. So It will not get distributed 100%. Like, for example, I took 100 drug, 75 reached my stomach, 50 got absorbed. All of 50, while being distributed, wouldn't reach to the target organ. It Five of it might go to my brain or somewhere else and show its effect, which we know as side effects. So that's why that bioavailability of a drug is important. So we can ascertain that what's the bioavailability of a drug by knowing is how much drug is received by the target organ as compared in oral route as compared to when taken in systemic or parenteral route. So that is how we determine the bioavailability of the drug. Now, there are various factors that can also affect bioavailability. First is the first pass hepatic metabolism, because when a drug is metabolized, most of it gets excreted through the kidney outside our body. So that's one of the reason, because when we take drug by parenteral route, it goes directly into our systemic circulation. But when taken orally, it gets absorbed, gets metabolized, distributed, and then shows its effect. So most of it gets released in the absorption and metabolization part. Then solubility of the drug, as mentioned earlier in the factors affecting that, if the drug is not soluble enough, then it's going to get excreted in its original form, and few is left to get absorbed in our body and be available for our target organ. Disintegration, also known as chemical instability, and nature of the drug formation. Liquid is absorbed more easily as compared to a tablet. Okay, liquid would be very easy to be absorbed in our body. Then comes the capsules, then come the tablets. There are other formulation, but I'm just comparing these three at the moment to make it easy for you to understand how the drug formulation affects the bioavailability in form of absorption. Now, bioequivalence. So there are two factors in the equivalence section. There is one bioequivalence and there is therapeutic equivalency. Now, if two drugs show the same bioavailability and peak time to reach, then it has bioequivalent. But if the drugs are pharmaceutically equivalent, they have similar clinical and safety profile, then it is therapeutically equivalent. Bioequivalence is when two different drugs are used for the same, like Nurofen and paracetamol being used for killing the pain, and they have the same amount being reached in the target organ and reach the peak concentration at the same time, then we can say, oh, they are bioequivalent. But when two drugs, two drugs are pharmaceutically equivalent, let's say paracetamol can be a branded one and a generic one. When these two brands make paracetamol 500 milligram, they are pharmaceutically equivalent because the ingredients same, they have same clinical profiles, they have same safety profiles. Both of them, if taken in high doses, can cause liver damage. Both of them has paracetamol 500. Both of them kills the pain, is an analgesic. So they are therapeutically equivalent. So we can use bioequivalent of a drug when we need to compare two different ingredients. But we use therapeutically equivalent, assess the therapeutic equivalency of a drug to understand the comparison between a branded and a generic. I hope I was able to explain everything today. And if there's anything that isn't understandable or still needs to be understood, or you want me to get more clarification, or if there's something that you want me to make understand upon such topics, you can let me know. Thank you.